problem? And I think the answer was -1/x squared. And I'm like, I don't know how to get there.
Yeah.
But I can type it into the calculator and do a secant or a tangent line. And it'll give me the equation for the tangent. So I could just type in the slope and it's the right answer. So, yeah. When it says solve algebraically, I'm just going to be like, I don't know. Yep. It is what it is. Got this.
You should get one of those TI-inspired ones that just do everything for you.
Okay, let's see here. Do I have, these tests are coming out, I thought I had them all, no? You guys still making paper airplanes out of them or something? All right. I'm gonna enter them in, if I don't have your test back, you can do it, right? Okay, so, I want to just cover like a short lecture content today to catch up on stuff. Again, we have no class on Thursday, and so we're going to start some lecture content. We're going to continue on the evolution, macroevolution topic with some stuff, and then today we're getting into analytics. We're going to do our first section, the first one section. Then next week, we'll do a deep dive into animal cells and tissues, and the fossil record, and all that good stuff. So I'll update the Canvas site. I haven't had a chance to show lectures and follow along with that review. But anyway, what we have going on here, though, is I want to talk a bit about So, I guess what I call this topic would be more evidence for macroevolution here. And one topic that comes up, especially with looking at the fossil record and genetic species concept, is homology. So we already talked about homology when we did our snail lab. We classified our snails that first week we had class, and I was kind of making this big thing about homologous or homology. And so let's go ahead and define that. That's a really important term or concept in biology here. But homology, obviously, ology means study of. And then the homo epithet here refers to the same. So what we're looking at is we're looking at the same structures found in different organisms, but oftentimes they can be used for different functions. So generally speaking, we have structures that have the same origin or the same common ancestry, but they might have been modified for a different use over time. So let's go ahead and say that really what's going on with homology is we're looking at common descent or ancestry. So homology infers that there was some common ancestor at some point in time. That's kind of the main point. So an example here would be the forelimbs of tetrapods. We have these different organisms. Humans. We have our forelimbs we use for grabbing and throwing stuff. Frogs, crawl or hop or whatever, similar to other amphibians. Bats use their forelimbs for flying. They have a wing that they suspend with their limb. A porpoise uses its forelimb for swimming. And then a horse uses its forelimb for running, obviously. So the main thing is these are different functional uses of the forelimb, but the limbs are made of basically the same parts. There's a humerus bone, a radius and an ulna, metacarpals, carpals, and phalanges. Basically, all these organisms have the same arrangement of bones. And so there's two possibilities. Either this is total coincidence, or there was a common ancestry among all the organisms. They got the basic structures and then later modified them. Interestingly, sometimes you can see that in homology, if you look closely, like for example, we have five phalanges or 5 fingers. Like in frogs, there's four externally exposed fingers. The 5th one is reduced, or what's called vestigial. It's a reduced finger. So sometimes superficially, homology doesn't look like it occurs. Sometimes you can find, evidence that there is a structure still present. Similar with the bat wing, it's kind of a weird coincidence that there would be 5 digits. There's like a thumb digit on the front that you use to kind of hang onto things, and then there's 4 additional digits they use to support the wing. Similar to the porpoise fin flipper is, we have a humerus, radius, and ulna, and if you look inside the flipper, even though it's a single kind of paddle, there are 5 phalange bones in the flipper. So again, these are kind of evidences for, is it coincidence, or is it because of some common ancestry, that the common ancestor of all these things had these arrangements of bones? The horse is kind of an interesting example of this, and it's wildly modified or wildly different in the toe arrangement. So we have a humerus, we have kind of a fused radius and ulna here, and then what amounts to the cannon bone or the lower leg of a horse is basically all essentially its middle finger. But sometimes you look at something like that and you say, there's no way we can make the comparison of this to these organisms. So sometimes with homology, you have to look at other developmental stages. So here is a drawing of a horse fetus development that by about 16 weeks or so gestation, the developing horse foal or horse fetus still has a humerus, a separate radius and ulna, and actually has 3 fingers. It has a middle finger and then the index and 3rd finger. That by about 6 or so months, those bones get fused, and those lateral fingers become the splint bones. The splint bones are bones that support the main cannon bone. So sometimes development, you can see the traits that are common, but then are later modified. So you're using these features that even though the adult horse has a super modified walking on their middle finger, and the splint bones are really reduced. If you look at the fetal development, you can see early on in development they actually have a similar feature of multiple toes and bone arrangement. And if you look at the fossil record of horses, and we're going to look at fossils next week, you'll see that in the fossil record, horses today, and for the last 20 million or so years, have the single toe feature. But if you go back to 35 to 55 million years, horses actually, the horse ancestors in the fossil record had multiple toes, going all the way back to kind of the stem horse or the common ancestor for horses, and that had 4 toes in the front, 3 in the back. So again, homology, we're using not only the traits an organism has today, but we're also using the developmental traits, we're using fossil record, all these things are fair game in homology. So that's kind of a common theme in describing macroevolution or describing how organisms are related, like we did with the snail shells, is looking for homology, okay? So anyway, again, sometimes it's difficult because you have, in some organisms, you have features that totally disappear. So like, for example, if you look at modern whales or dolphins or porpoises, they have their foreflippers that we described with the 5 fingers, but their hind limbs are drastically reduced. The hind limbs are basically reduced down to just pelvic bones. They're suspended kind of in the rear portion of the body. Pelvic bones are probably still supporting, some muscle attachments, but there's no external limbs at all in these organisms. Instead, they use their tail as a propulsion device. But if you look at the fossil record of whales or dolphins or what have you, going back 30 million years to 50 million years or so, the fossils show whale-like ancestors that had hind limbs that were exposed. And occasionally, I think I've seen pictures like from the whaling days of sperm whales with like hind limbs, like baseball bats sticking out, kind of like a birth defect essentially, showing a primitive trait and showing out. So again, the idea is vestigial traits can sometimes be deceiving in that you can't see homology because it's a reduced trait, because it has reduced function, or what have you. Okay, so the complicating factor is sometimes a trait that is used for the same function can fool you into thinking that there's common ancestry. So the opposite of homology or homologous traits is what's called analogous traits. So an analogous trait is the opposite. It's a trait that's used for the same function, but it comes from different ancestry. So if we look at organisms that fly, we talked about the bat already. It had a humerus, radius, ulna, and it has 5 phalanges, 5 finger digits. Birds also have a humerus, radius, and ulna, but the arrangement of the wing is different. They don't use their fingers to support the wing. They use scales or modified scales, feathers, which are modified scales, to support the wing. So, we'd say that because the arrangement of bones is so different, the common ancestor of bats and birds, which probably occurred, somewhere in the 250 years ago, didn't fly. So there was no common ancestry of flight between these organisms. They independently developed the ability to fly, and that evidence is that the bone arrangements are sufficiently different enough. Certainly, insects that learned how to fly upwards of 400 million years ago have a completely different mechanism for flight. Their wing is basically a modified gill structure. So again, this is what's called convergent evolution. It's where you have traits that are for the same function, but they come from different ancestry, and they can often confuse classification, like we saw with the snail shell lab. All right. So, on the exam, this exam, I'll probably have, I really like compare and contrast questions where you compare things. And on this exam, I'll probably have like a list of compare and contrast, like compare and contrast homologous traits versus analogous traits, right? And that would be an example of, okay, here we have how they're similar is their adaptations, how they're different is homologous traits are from the same ancestry but different function. Analogous traits are the same function but different ancestry traits, not the opposite. And then I'll probably have vestigial structures as a comparison to, we'll see some another term come up here called exaptation. I'm not going to define it right now, but we'll see it as an example in class. But I'm going to probably come up with some of these terms. These terms are kind of used commonly in zoology. With that being said, let's take a look real quick here at some species concept issues. So anyway, for the last exam, I was emphasizing that the biological species concept is still the most widely accepted and widely cited species concept. The idea is is you're a member of the same species if the population can interbreed and produce viable offspring. Right? So it's kind of like looking at whether populations of the same species are different. I mentioned there's a bunch of problems with this, namely that there is most species are asexual. Oops, we can't really use sexual reproduction as an example. And I also mentioned that we have things like fertile hybrids like polyploids. So the biological species concept is not perfect, but it's still the most widely cited species concept today. There are some alternatives out there though. One of them is called the evolutionary species concept. And where this comes from is paleontologists. When paleontologists are looking at the fossil record, obviously they can't apply the biological species concept to fossils. So the paleontologists basically look at a species as a ancestor-descendant lineage of populations that maintains its identities from other such lineages. It's kind of a vague concept. It's hard to really pin down or test. But I want to point out, I guess the test question will be, why do paleontologists need an alternative species concept from the biological species concept? And the answer is because fossils, are remnants of ancient organisms. You can't apply the biological species concept. So paleontologists use an evolutionary species concept, which essentially is just like Linnaeus or Darwin would have done, right? Typological or nominal species concept. Paleontologists are essentially using the same idea. So I want to point that out, that the biological species concept is difficult to apply to many circumstances, certainly to the fossil record. One more idea here that is today probably the most rapidly developing and probably the most cited species concept. I don't have a slide that defines it, but I'll talk about it during class, is what's called the genetic species concept. Genetic species concept. Okay, and this is over the last 20 years has become probably as popular, if not more popular, than the biological species concept. And the reason is now it's relatively easy to collect DNA from organisms and do some sequencing of DNA of those organisms. So anyway, the genetic species concept basically works off of divergence. How genetically different are two organisms. And so where this comes from is when the Human Genome Project was wrapping up around the same time. So the human genome was entirely sequenced and decoded essentially by the early 2000s. Around the same time, other organisms were being sequenced and figured out at the same time. And what was determined was that, for example, humans so human versus human comparisons is humans are about 99% similar. So there is less than a there's like less than a 0.5% difference among all the humans on the planet. So if you compare, say for example, Native American populations with native populations from Africa or from Australia or what have you, among all humans there's less than 0.5% difference in the DNA. So this is a DNA difference. And then when they looked at human chimpanzee, which would be presumably the closest species to what humans are, a primate that would be the closest to human ancestry, for example, there is approximately a 2% DNA difference. So, because, we like to describe ourselves and talk about ourselves, this 2% has been used as sort of a threshold or benchmark for if you have greater than a 2% DNA difference, or 2% or greater DNA difference, you're considered separate species. If you have less than a 2% difference, you're considered the same species. And so so the main thing to think about the genetic species concept is using DNA sequence differences. So when you're looking at, say, for example, the gene for hemoglobin, like the gene for the hemoglobin blood protein, you'd imagine that organisms that had a more recent common ancestor would have more similar DNA code. And because mutation kind of happens at a constant rate, that the older or the farther ago your common ancestry was, the greater the difference in DNA. And so that makes sense. And the other reason why this is popular is because it's so easy now to get a DNA sequence. It costs like $15 to get like 1,000 base pair sequence, and you can get the data in like less than a week. So as you know, with 23andMe, basically that's what they're doing, is they're using DNA sequences genes in your home gene testing stuff. But anyway, what I want to point out is this 2% is really human or primate or certainly mammal specific. So I have an article on our Canvas site. Let me just show you real quick here. Close up here. Let me get this going on here. And let's see here. Okay, so we'll take a look at lecture exam sheet content. Genetic species concept. So this is a really, so there's some papers in here that I want you to kind of pay attention to. Again, you don't really have to read the whole paper. You can kind of look at the abstract and kind of skim some of the articles here. Let me see if I can get this open up here. So anyway, this is an article about comparing the DNA sequence of a rodent species. So they use rodents as their model for whether organisms were the same species or different species. So what they did was they kind of compared the biological species concept, whether those organisms interbreed, and then they looked to see if the DNA reflected this 2%. So you can see here, let me go ahead and expand this out here. You can see here that the 2% was kind of a target they were looking to see if this made any sense. But the problem was, in these rodents, they found that populations could have as high as like an 11% difference and still interbreed and produce fertile offspring. So the issue here is that when you look beyond the human model, the human chimp model, that rodents basically could be as much as 10%, 5 to 10% difference. This is all kind of, this is not even in your book, this is kind of stuff. So the rodents, according to this paper, there could be as much as a 5 to 10% difference, and they still acted like the same biological species because they interbreed and produce viable offspring. Birds and reptiles could be anywhere between 10 to 15% difference. And invertebrates like insects, could be like 20% difference. So the main point here is when comparing DNA between organisms, to use that as justification for calling them separate species, the same species, there is no one number that can be used universally across all species. 2% is used as a general rule of thumb only because the human and chimp genomes were one of the first genomes described, and that 2% difference was documented and reported. But it appears that other animals, could be range even as much as 20% difference and still produce viable hybrid offspring that were fertile. So I guess my main point is, for the test, is on a genetic species concept, the 2% difference is kind of our rule of thumb, but it really only applies to primates or the human chimpanzee model. The range is so great that no one rule of thumb number could be applied. So there's not like a threshold of DNA difference. The issue is because DNA is so easy to sample and to get a sequence from, it's really popular. So just be aware of that. On the exam, I'll probably have an example of some article or something there that mentions some DNA differences between organisms. I think last year I used bears, like black bears, grizzly bears, and polar bears. Those are the three main bear species that we have in North America. And the article basically did a DNA percent comparison among bear species. And the main thing I'm looking for is that you reflect the idea that the genetic species concept uses the 2% as a threshold, but other mammals that are not primates could be as much as 5 to 10%. So the answer is not totally clear in terms of what a useful threshold for DNA difference is across all species. So that being said, let's take a look at our other things we have here. There's another article here that's interesting, and these are papers or articles that I want you to kind of look at. This is kind of your homework for Thursday. For not being in class Thursday, I want you to kind of just at least open those up and take a look at them. Where the DNA percent difference comes from is from mutation. So here is an article that's really interesting that is basically goes through the idea of a background mutation rate. There is a natural kind of mutation that happens from generation to generation. So genes get modified, but basically new alleles or new traits arise from mutation. And this mutation that when populations don't interbreed results in the difference in DNA. So, scan the introduction to this article, but basically it summarizes essentially what the background mutation rate is and how that mutation rate can be used as some measure of evolutionary time. Okay, and then there is another article here that's really cool on equine evolution or horse evolution. And this is fun because it actually goes into populations of equines, zebras, asses, and wild horses. And it actually goes through and does a chromosomal DNA difference here. So in this article, here it is. This is the cool image. in this article, it basically goes through and describes among all the different equines, using domestic horses as kind of our root comparison. It looks at the chromosomal and DNA differences and tries to project how long ago those populations occurred. So for Thursday, I'd say we have no class, but at least open these articles and check it out, because they're all reflecting this idea of the genetic species concept, which when you transfer to graduate school, you probably will do something regarding DNA or genomes for your organisms that you're working on, because it's just so popular. Okay, any questions we got? So that's all I was going to cover for lecture today, which is homology, analogous traits, genetic species concept. The evolutionary species concept, just think of that with regards to what paleontologists have to work with to identify species, and that's the main idea. So let's just go ahead and jump right into the lab here. It's not that challenging for lab today. Today we're going to look at our next phylum.